like this, keep this recording throughout the class. Thank you everybody for putting your name in there for checking in this time around. I'm going to try to go through this material fairly quickly so that you guys have plenty of time to work on it this evening, ask questions if you need, and we will go from there. So I'm going to try to share my screen. Can you guys see my screen now with Canvas pulled up here? Let's get rid of all these.
Yes.
Excellent. Okay, so today we are starting lab 3. So for lab 1, safety in the microbiology laboratory, your quizzes should be completed. You guys should have the scores for those quizzes available to you now. For lab 2, those quizzes were due last night. So I will try to grade those quizzes by next week, Tuesday. But it is a holiday, so I don't guarantee that I will get it done by next week, Tuesday, but I will try to. So those will be probably available by then. So we reviewed microscopy last week, bacterial cell structure, and the 2 different types of staining that we looked at, negative staining and gram staining. And do anybody have any questions on that material right now? I'm going to refer to it a little bit, but if you have any specific questions, let me know.
No questions.
Excellent. The first document we're going to take a look at today, so that just as an overview, we're going to go through acid-fast staining, so a 3rd type of staining, and then we're going to spend quite a bit of time on growth factors, and then talk a little bit about bacterial isolation and the methods for doing that. So the first one, if you guys want to follow along, acid-fast staining. Let's see, make sure my recording is still working, which I believe it is. Oh, that was weird. Sorry. Ha, weird. Anyway, hopefully that's still working. And so acid-fast staining. So the 2 types of stains that you guys already encountered, sorry, I'm using a different computer, I apologize, are the gram stain and the negative stain. So the very first thing it says here is acid-fast stain is a differential stain. What exactly does that mean? So you guys have already encountered 1 type of differential stain, and that is the gram stain. A differential stain is a stain where essentially you can differentiate between 2 groups or 2 types of bacteria or really anything, and tell the difference between those 2 groups. So the gram stain is differential between bacteria that have that peptidoglycan layer on the outside and the bacteria that have that double membrane structure with a thin peptidoglycan cell wall in the middle. That's what that differentiates, the gram stain. The acid-fast stain differentiates between bacteria that contain mycolic acids versus bacteria that do not contain mycolic acids. And we're going to talk a little bit about those. So, yeah, so an acid-fast stain won't tell you anything about whether or not a cell is gram-negative or positive, and a gram-negative or a gram stain won't tell you anything about whether a cell is acid-fast positive or not. So they're 2 completely different tests, and you can't infer anything from one to the other. The reason why the negative stain is not a differential stain is that the negative stain is essentially staining the background of the cells. It's not actually staining the cells themselves. So you're not differentiating between the types of cells with your stain. The negative stain is repelled by the charge on the cells, the teichoic acid and the lipopolysaccharides on your gram negatives and positives. They repel that stain, and it essentially then just stains the slide itself instead of staining the bacterial cells. So negative stain is not differential, gram stain is differential, and acid-fast stain is differential. Because it is detecting the presence or absence of mycolic acids. These are considered a virulence factor. So that's what this is talking about here. Mycolic acids are a virulence factor because they are basically what causes these cells to be able to cause disease. It makes them difficult to treat, so that is considered a virulence factor, and you guys can read a bit about that here. The only two genera of bacteria that have mycolic acid are the Mycobacterium and the Nocardia. If you remember, we talked a little bit about Mycobacterium in that Mycobacterium tuberculosis is the source for TB infections or tuberculosis infections. But there's lots of different types of mycobacteria. And so because the acid-fast stain detects any and all mycobacterium, it is only a presumptive test for tuberculosis, essentially, is what you might use it for. If it comes out positive, which would be pink, you might suspect that a patient who has acid-fast positive bacteria in their lungs has TB, but you wouldn't know it unless you did further testing. So that's what they're referring to down here, presumptive testing. So you can read about the different stains used in the acid-fast stain. The primary stain, like the Gram stain, there's a primary, there's a counter stain, and there's a decolorization step. The difference here is the Gram stain also has a mordant, which is that iodine, which sets the dye. Carbofusion does not use that a mordant. So definitely read through this, become familiar with the process for acid-fast staining, and I believe there's a video on how to do the acid-fast stain. So I just wanted to share with you guys this photo and just talk a little bit about what you would see in an acid-fast stain. So the Gram stain, you're looking at pink and purple cells. Oh wow, this is not set right. For the acid-fast stain, oops, that's not gonna do it. For the acid-fast stain, hopefully you guys can see my mouse because my little pointer thing is not working. These little pink cells here would be your positive cells. So most of this is acid-fast negative, which is all of this blue area in here. But the little pink cells in there would be what would be considered positive. And this would be acid-fast positive test if you were going to be running this test. Okay, so the pink rods, acid-fast positive, blue Kasai, acid-fast negative is what this is showing. So definitely read through that, become familiar with this particular stain, and then answer these questions for the Dropbox. And that will be that section. Oops. So going back to, so remember, here's our Dropbox. Again, this is going to actually be due in, let's see if I can open a calendar here. Sorry, this is not my normal computer. So that will be due technically the 22nd by midnight because you guys do not have class next week. But it's probably better to get this done sooner rather than later. So excellent. Next one we're going to be looking at is the growth factors. So let's close that one. So there's quite a bit of information in this particular document. As you can see up here, it's actually 12 pages long. So when you guys are doing this, just prepared that it is quite a lot of information and definitely worth taking your time to understand. But I think once you guys understand it, it's a pretty useful concept to know for microbiology. So we're going to actually look at 3 different growth factors. And when I'm talking about growth factors, I'm talking about how bacteria or microbes like to grow. So there's different things that influence how they grow and influence where they can grow. So in our first lab, we talked about how microbes were ubiquitous. They grow everywhere, but different bacteria have different adaptations to grow in different locations. So we're going to look at 3 of these adaptations. We're going to look at temperature, oxygen, so whether or not bacteria use oxygen, and osmotic pressure or water activity is the third one. And all 3 of those are different sections in here. So we're going to start with temperature. And there's a lot of terminology in here, so I'm going to be trying to kind of show you guys how I would approach learning all the terminology and different ways of learning that. So, but for temperature, you can see here, bacteria have what's called a growth curve, or and this labels it as a growth scale. So the growth curve of a bacteria shows the minimum temperature at which it will grow, the maximum temperature at which it will grow. Any higher than that, it would die. Any lower than that, it would die. And bacteria have an optimal temperature, so they can sort of grow in some of these areas, but they really like growing in certain areas. So I kind of refer to it, I kind of like to think of it in terms of humans. We have a minimum temperature that we'll live and survive in, so we can survive in Antarctica, but we might not like it. We can survive in Las Vegas and it's the middle of the summer, but we might not like it. But we're really, really happy when we're on the beach in LA and it's, 70 degrees. So that's like our optimum temperature. So bacteria are the same way. They have a minimum, they have a maximum, and they have an optimum. The thing to understand about this curve, so going back to my board here, let's see if I can make this full screen for you guys, is that these curves can look a little bit different depending upon the type of bacteria. So apparently this is not going to let me put my mouse over. I can share this with you guys with my other mouse. Okay, so the one we were just looking at looked a little like this. Sorry, this is going to look really bad. So there was a minimum, there was an optimum, and there was a maximum. But these curves can also, so if there is a cold-loving bacteria, so this would be like what we'll call a mesophile. We're going to talk about these terms. So they like that Goldilocks zone. They like those middle temperatures. There could be something called a psychrophile, which likes cold temperatures, and its growth curve might look like this, where its optimum is at 0 if this is 0. And its minimum is down here somewhere. Where'd my mouse go? I've just freaked out the computer. Or can you still hear me?
Yeah, I can hear you in here.
Sorry. I'm using my husband's computer and it just kind of freaked out on me. So yeah, so we were talking about temperatures. So this is what a psychrophile would look like, or a cold-loving bacteria. Their minimum would be below 0 maybe. Their maximum or their optimum would be maybe at 0, and their maximum would be, maybe lower down here. So you can shift this curve left or right. So something that's maybe an extreme thermophile might look something like this, where you move that curve over in this direction. It locked me out and then tried to sign out and then did some weird things. Okay, sorry guys. So this is what so when we're talking about these growth curves, they're different for each species of bacteria, and each species of bacteria will have its own individual growth curve. So let's see. Let's go back to our other screen here somewhere. Yeah. So that is what this is showing here. And then some other terms it's talking about here are absorbance and optical density, for how you actually measure or draw these growth curves. So I wanted to talk to you guys a little bit about that and what that means. So if you, I'm going to go to my next figure here. So we're talking about absorbance and optical density. So the way that you measure the growth of bacteria to figure out their minimum, their maximum, and their optimum, this would be your eyeball. Sorry, my pen is not working. So a high absorbance would be if there was a lot of growth in your tube. So if there's just a lot of bacteria growing for a long time, it gets super cloudy. The light that would be shining through is going to hit that, and not very much of it is going to make it through to your eye, or in this case to the receptor on a spectrophotometer that reads absorbance. So you're going to get a high absorbance value right here because not very much light gets through, so most of it is absorbed within the sample or reflected back from the scanner, the spectrophotometer. In a low absorbance culture, you would have, just a little bit of bacteria growing. Maybe the culture is not very old. Maybe it's dying, but for some reason, or maybe it's just not at an optimal temperature. You would just get very little growth and light shining through that. Most of the light is going to make it through. Some of it's going to stop and be reflected to your spectrophotometer or your eye, whatever you're using to receive the light, and that's going to register as a low absorbance number. So if you look on here, this is absorbance from low to high. So the higher the absorbance, the more optimal or the faster that culture is growing. And you get a cloudier culture, a more turbid culture is the word, and you would get a high absorbance reading. So if you chart these absorbance readings, you can tell where the culture dies and where it's growing optimally. And that's the next section there. Okay. So that is temperature. And there's a bunch of terms associated with temperature bacteria that you guys need to read through and learn about and learn their temperature ranges. But there's a lot of these words. There's psychrophile, psychrotroph, mesophile, thermophile, extreme thermophile, and thermoduric. So how I would learn these terms, let's talk about these a little bit. I just wrote up a quick chart based on all of those terms that were in there. And so when you're looking at these terms, you'll notice that they have two different portions. They have a prefix and a suffix, basically, a beginning and an ending. And you can mix and match these terms and basically form new words. So, for example, in that document, there's psychrophiles or psychrophilic bacteria. So anything, let's look at the endings first here. Anything that ends in phile or philic is referring to loving. So if you think of the word Philadelphia is the city of brotherly love. So philo or philo is referring to love. And in bacteria, this means that they require whatever it is you're referring to grow. They require it to grow. It can also end in troph or trophic, and that's just talking about it living. Duric, it could end in duric, so it could be psychroduric. That could mean that it survives, but it doesn't grow. That would be what it's referring to. It's going to persist in some, usually spore form, but it won't actively be growing. And then there is tolerant bacteria, so you could have psychrotolerant bacteria, and these are bacteria that don't require whatever that is, like the philic bacteria do, but they can survive it and they can grow in those conditions. So they are tolerant of it. So psychro, does anybody know what psychro refers to? You can type it in or tell me. So psychro Anybody? Anybody? Everybody left. Psychro would refer to cold. So these would be bacteria that like the cold. Meso is referring to those middle temperatures or that Goldilocks zone. So they just usually prefer stuff like room temperatures. Thermo is probably pretty obvious, but it's referring to heat. So they could be heat-loving or heat enduric. Thermoduric would be surviving high heats. Halo, we're going to talk about this next. Halo is referring to salt, and osmo is referring to osmotic pressure. But in bacteria, it's typically referring to a high sugar environment, or sugar or solute environment. And finally, the third one we're going to talk about after we talk about water activity with halo and osmo, we're going to talk about oxygen, and that would be what aero is referring to in requirements. So you could have something that's aerophilic, aerotolerant, osmophilic, osmotolerant, and that's how I would try to learn these particular terms throughout here. So the next section in here we're going to be looking at is the scroll down here. There's some results, some formulated results from an experiment that you guys get to process and write out and interpret. So make sure that you're able to interpret temperature curves. The next one is going to be water activity. So water activity is the availability of water for a bacterial cell or for anything. And if the water activity varies between 0 and 1, if it's at a 1, there's pretty much pure water. And if it's at a 0, it would be pretty much, the water would not be available at all. It would be very salty or something like that. So depending upon the water activity of the substrate that the bacteria is growing in, such as high sugar environments can have a very low water activity, or high salt environments can have very low water activity. Bacteria need to be able to adapt to those conditions. And one of the ways that they adapt is by changing their lipid membranes to resist lysis, or to resist extreme heat or resist extreme cold and stay flexible. So there's various ways that bacteria can modify themselves evolutionarily in order to adapt to these extreme environments. So we're going to talk a little bit. I wanted to go through with you guys osmosis. It's a thing that seems to hang students up a little bit when we're talking about this. So I'm going to go back to my pictures here. So osmosis, we're talking about water moving from low solute concentration to high solute concentration. So if, I'm just going to abbreviate because I can't write apparently. So if there is salt in the cell, which there's always salt in the cell, because there's a lot of cellular components, and this is our cell here, and if there is also salt in equal concentration outside of the cell in the environment, say on your skin where your skin is salty, if these are equal, water will flow equally between the cell and the environment. You won't get water flowing one way or the other. This would be considered an isotonic environment. I'm going to type, this is ridiculous. Isotonic. Where that's equal flow of water into and out of the cell. So in this situation, the cell would be potentially perfectly happy. There's no adverse conditions going on for the cell because everything is equal. Where you get different things would be in a hypertonic solution, where there's greater salt or solute inside the cell. Let's see. So because there is greater salt outside of the cell, water always moves from low solute concentration to high solute concentration. So this is going to actually pull water from the cell to the outside of the cell. And some bacteria can survive it, but a lot of bacteria, what happens here is all of the water leaves and it's like a balloon. The balloon just collapses. And you end up with kind of a cell wall that the bacteria have, those peptidoglycan layers, but all the insides of the bacteria kind of collapse and the bacteria dies. So this can result in death of bacteria. Let's put a big X on that. If all the water leaves and the bacteria doesn't have the adaptations to resist that. And then the opposite of a hypertonic solution would be a hypotonic solution. So there's less salt, or solute, I'll just say, because it can be sugar, salt, anything like that outside the cell. So this would happen if you maybe threw bacteria into DI water where you've removed all the salt from the water or something like that. So a hypotonic solution, water would want to be rushing into the higher salt concentration of the cell. So you would potentially have, let's change colors here. You would have the water going from outside of the cell into the cell. And if the cell is able to keep its cell wall intact, it just gets all plumped up and potentially happy. But most cells cannot handle a flow of water into the cells. And what happens for them is they actually get really plump and then eventually they burst and everything leaves the cell and the cell dies. So this also ends in cell death. So yeah, so it basically breaks open a hole in the wall of the cell and all the cell contents spill out in a hypotonic solution. So these are different adaptations that bacteria have to develop to live in very salty or very sugary environments are the two that we're going to talk about. Cool. So yeah, some of the terms that you'll see in here, hypertonic, hypotonic, isotonic, we just talked about those three terms. Plasmolysis, that's where the cell bursts open. And then halophiles, we talked about halo meaning salt, phile meaning loves or requires. So a halophile would be a bacteria that requires a salty condition. And there's slight, moderate, and extreme halophiles. There's bacteria that are halotolerant that don't require salt because they're tolerant, but they can persist in a salty environment and survive and grow. So that would be a halotolerant bacteria. And then there's osmophiles. Those are referring to those sugar-loving bacteria or those sugar high concentration, high osmotic pressure requiring bacteria. So again, there's another example of an experiment, of the results of the experiment. Just know when you're looking at these results, your lines are your drawn on black lines. The growth is these white areas in here, actually variously colored, but that's where your bacteria is growing. So these are actual results to this experiment. A table to fill out for that experiment, as well as questions to answer about that experiment. And finally, page 9, oxygen requirements, aerotolerance. So aerotolerance, again, referring to oxygen and whether or not bacteria can tolerate and grow in the presence of oxygen, or if they cannot tolerate and cannot grow in the presence of oxygen. So oxygen is a really great electron acceptor for producing energy, or ATP, probably what you've learned from a basic biology or biochemistry class. But oxygen can also produce toxic metabolites. So those oxygen species that are formed during this process are very poisonous to cells and cause a lot of damage. So you need to be able to get rid of them in order to survive breathing oxygen. So the area I wanted to focus on here, so make sure you guys are reading all of this, understand it. This little chart is very helpful, and I wanted to go over a couple of them that are confusing to people. So let me zoom in here a little bit so you guys can see that a little better. So for our, a couple of these are pretty straightforward. Number one, strict aerobes. Those are bacteria. As you can see, they're growing on the top of this culture. They're interfacing with the oxygen that's in the environment. So they're able to access that oxygen, and that's where they're going to grow because they require oxygen. Aerobic bacteria require oxygen. Next one, strict anaerobes. These are bacteria that cannot survive in the presence of oxygen. So they're growing on the bottom of this tube because the oxygen from the environment is not going to diffuse down to the bottom of this tube. So that's where you have your strict anaerobes and your strict aerobes. The next one that's pretty easy is number 4, are your microaerophilic bacteria. These are bacteria, so let's break down this word. Aero is oxygen, philic would be that they require oxygen, but they require small amounts of it, microaerophilic. So they can't, or they don't like to grow at the very top of the culture where there's a lot of oxygen. They like to grow in a slightly lower layer where there's minute amounts of oxygen. Do need oxygen to grow, so they cannot grow at the bottom of the culture. And then the two that people always confuse with each other, so definitely easy to quiz people on, are number 3 and number 5. Number 3 is facultative anaerobes. Number 5 are aerotolerant. So anaerobes, again, don't require oxygen, but these are facultative anaerobes. So these bacteria can use oxygen and can grow in the presence of oxygen, but in the absence of oxygen, such as the bottom of this tube, they can also grow, and they do it by switching systems in which they're producing their energy. So they'll usually switch from oxygen respiration to fermentation. So an example of a facultative anaerobe would be yeast. Yeast can happily grow in oxygen. It's how we make bread, those types of things. But yeast can also switch to fermenting if it's in a no oxygen environment, and we can produce things like beer and wine. So yeast can switch between using oxygen and not using oxygen, and that makes it a facultative anaerobe. Aerotolerant bacteria, on the other hand, do not use oxygen, do not require oxygen, do not use oxygen, but can tolerate it. So they're a little bit more on the side of anaerobic bacteria, but they can tolerate oxygen, and so they can pretty much grow anywhere they want to in this column, in high oxygen, in low oxygen, and no oxygen. They can tolerate the oxygen, but they don't use it for growth. So those are the 5 different types there. Again, make sure you're reading through all of this. I believe there's another experiment down here that shows growth of bacteria in oxygen, so O2, and with no oxygen, no O2. And how to interpret those plates and interpret the results from those plates. So there's quite a few questions to answer for this particular document. Make sure that you're turning those in into this growth factors Dropbox. And then the final one is for your simulation in Labster. It's a pretty fun simulation. where you get to isolate some bacteria, and it is, again, this is the lab manual for the labster, so you can either read through this, learn a lot of the information, but you can also learn a lot of this in the simulation itself, so I'm not going to go through it a lot. I just want to point out definitely things that you will be, tested on in the future is going to be streak plating, so learning how to do streaking. And there's some different diagrams here for the first streak, the second streak, the third streak, and you need to learn about how you need to sterilize your loop between each and every one of these streaks in order to obtain individual colonies of a species. So that's pretty much what that entire simulation is about, but definitely an important concept to understand by the time you're done. They're also going to hit on sterile technique, making sure that you're doing this in a sterile or aseptic environment, so that you guys are able to, so that when you're working with these things, you're able to come up with those microorganisms that you're looking for. And finally, they talk a little bit about linear versus logarithmic scales. Just know that when you're talking about the concentration of bacteria, because bacteria grow pretty much exponentially, so they separate into 2, and then those 2 separate into 4, and those 4 separate into 8, etc. You get exponential growth in bacterial cultures. So in order to visualize that on a chart, you need to draw it in a log scale rather than a linear scale. So these are both the same thing. But as you can see here, this linear scale goes from 0 to 10, whereas this linear scale can go much higher in the same amount of space, and it's just log transformed so that you can visualize what's going on there. And it just notes here that it's a pretty common scale. If you've never encountered it before, it's used in a lot of different scientific applications, but growth curves, bacterial growth curves are one of them. Yeah, so I did want to hit on bacterial isolation. So we talked about the 3 different growth factors: temperature, oxygen, and water activity. There's a 4th growth factor, so those are things that can impact how or where something grows. There's a 4th type of growth factor that I want to chat about, and that's the media itself that you're growing the bacteria on. So there's 4 different types of media that we'll be talking about off and on throughout the semester, and each of these selects for different growth of bacterias in different ways. So the first one is differential, and as you might surmise, this is very much the same as differential for our staining purposes, and this is just the media that you can tell the difference between 2 different. Let me just type it in RNs. It's usually a types or species or genre, and it really depends on the media. So one of the examples of a differential media that you're going to encounter in this lab is for Salmonella E. coli plates, where you can tell the difference between a Salmonella colony and an E. coli colony on differential media. And that's one of the ways of selecting and isolating specific types of bacteria. Another type of media is selective media. So selective media actually inhibit. So the important thing to understand between selective and enriched is selective media inhibits some bacterial growth and promotes others. An example of a selective media would be a dextrose media. that would prevent or inhibit the growth of bacteria in one of those high osmotic pressure conditions and promote the growth of fungi, because fungi are a little bit more burly and can survive some of those high osmotic conditions that we were talking about before. Enriched is the third type here. Enriched, kind of as it refers to, is where you're adding ingredients that promote the growth of a certain type of bacteria. And one of the examples of this would be something called a blood agar plate, or a plate that has blood cells in it, and that promotes bacteria that can utilize the iron in those blood cells. So that promotes the growth of those specific types of bacteria versus other bacteria that can't access that iron. So that's another type of way of differentiating and enriching for certain types. And the last one here, pretty self-explanatory, is general purpose. Pretty much everything on these plates. And that's just if you want to see, if you're taking an environmental sample and you want to see if there's anything present, you might use a general purpose media. So those are the 4 different types of media. You're going to be, encountering several different types in this simulation, so keep an eye out for those. Try to differentiate between what those are. I think that was pretty much all I had for you guys tonight.